Okay, now let's talk about one of my favorite topics in that unit: transmission, infectious dose, and basic reproductive number. My notes aren't updated, and I know that, but I'll give you some kind of new ideas. They want these new ideas. I won't ask them on the exam specifically, but they like real life medical concepts. So, infections overall, in terms of the transmission, can be communicable and non-communicable. Does that make sense? Now, examples of communicable infections are plentiful, obviously, from influenza to gonorrhea to HIV to coronavirus to norovirus. We can go on and on and on. Now, what about non-communicable infections? There's not really like a general, how to say, concept of what can result in non-communicable infection. But again, generally speaking, in the humans, in terms of transmission from person to person, infection of central nervous system are non-communicable. Does that make sense? So if a pathogen in the brain, it cannot get anywhere. Now think about it. You don't have a portal of exit when it's in the brain. Remember we talked about brain-eating amoeba. It's stuck there. It can't escape. Am I clear? You can argue. Now what about polio? It causes the infection of CNS. Yes, but the virus that's in CNS isn't getting transmitted. It's the virus that's in the gut that gets transmitted to other folks. Does that make sense? So you may have heard about Legionella, legionellosis. Legionella pneumophila infects macrophages in the lungs. You acquire it from the environment, and that's it. Clostridium botulinum is non-communicable infection. Tetanus is non-communicable infection. You can't transmit it to other people. Does that make sense to you? Which brings us to a very vaguely defined the concept of contagiousness. Okay, and this is actually like a really nice conversation that we're going to have. So contagiousness. How many of you have heard this frightful screams about how contagious the Delta variant is? I guess many of you, yeah. It's like all over the news. That's the only thing they talk about. It thankfully takes away the attention from another Delta, which is an airline company. So contagiousness. People say, oh, more contagious, less contagious. Can you measure this? Technically, yes. You can measure this using basic reproductive number. Basic reproductive number, or R0. Okay, so this number estimates how many people will get infected from a single infected person. Does that make sense? So you have a fella here that's infected. Okay, this fella is infected. Let's say this fella transmits the disease to 3 more people. Okay? Does that make sense? In this case, R0 is 3. Are we clear? So a few things. First of all, basic reproductive number, or R0, characterizes only the ability to transmit, not the severity of infection. My favorite example, Ebola versus measles. Remember we were talking about it. Ebola, R0 is about 2. It is really poorly transmitted. Measles, R0 is about 15 to 18. Most transmissible disease known to humans. Measles. But if you would ask me what I would prefer, measles every time of day. You see, it's not a severity. Does that make sense? Good? Now, can you measure R naught? Technically, no. You have to model it. You have to measure the incidence of infections. You have to, test people. You have to look at the interactions, and you have to apply certain models to calculate it. Does that make sense? Calculating basic reproduction number is incredibly hard. This is why when you hear, oh, R naught for this variant is 7, bullshit. Let me explain why. Because what people measure actually, what epidemiologists measure, is effective reproductive number. Imagine for a second, and I want you folks, you go into the healthcare. I want you to be educated and how to say, what's the good English word? Knowledgeable in this matter, because it will guide your decisions as healthcare providers. So imagine that we all infected with coronavirus, okay? And we decide, here's what we're going to do. We're going to go to massive parties and spread it as much as we can. In this case, the effective reproductive number will be probably like 30 for that situation. Does that make sense? On the other hand, if we decide, okay, we're going to wear, the entire population is going to wear masks, we're going to limit the assemblies of people, we're going to sit far from each other in the class, and so on and so forth. We essentially reduce the chances to transmit. Does that make sense? So effective reproductive number starts to decline. And the whole idea behind this masking and reduced interactions and so on and so forth is to bring effective reproductive number below 1. Because if one person can transmit virus to less than one person, eventually the infection will die out. Does that make sense? So this is something like really important to think about. And this is why the whole idea of Delta variant being so contagious is bullshit. For viruses, it's not transmissibility. It doesn't mean that it penetrates the mask. It doesn't mean that it hops over those plexiglass boards. It doesn't mean that it just flies on on the wings, like 50 yards from where you're breathing. No, it's all wrong. For some reason, this virus is evolutionarily more fit. It has higher fitness. Maybe it's replication fitness. This is why it takes over. Am I clear? That's number one. Number two, look at where all the new infections happen. They happen in the pockets with the low vaccination rates. That's it. Period. Where people don't get vaccinated, they get infected. They get infected, they spread it to other people. And last but not least, but that's like my tiny little pet peeve, everybody reports new cases, but everybody conveniently avoids reporting new hospitalizations and deaths. And they do not climb up. They really don't. Vaccine protects against hospitalizations and deaths. Yeah, I'm vaccinated. I get infected, well, it's inconvenient, but I'm not going to die. I'm not going to end up in the hospital. Maybe I'll get a sniffles, something like that. Are we clear? That idea. Okay. So this R naught story brings us to the conversation about the infectious dose. Okay? Well, infectious and lethal dose. So we have two terms here. One of them is ID 50. Median infectious dose. Okay? So this is the amount of virus that is needed to infect 50% of the exposed cohort. That makes sense? And then we have median lethal dose, LD50. This is the amount of virus or bacteria or whatever pathogen you're talking about that is required to kill 50% of the inoculated cohort. Does it make sense to you? So median infectious dose and median lethal dose, first of all, are insanely hard to measure on humans. Let me explain. I can take several groups of mice, give them different dilutions of a virus or of bacteria, and then observe how they die. Am I clear? I can measure very, very precisely median lethal dose or median infectious dose on animals. I can't take groups of people and expose them to a virus in a controlled environment expecting them to die. It's unethical, right? This is why for people, for humans, it's usually estimates. That's number one. Number two, infectious doses differ dramatically. Number three, and this is probably the most important, you cannot compare between different pathogens. You can compare taxonomically similar or taxonomically close pathogens. Couple of really, really striking examples. The infectious dose, have you heard about norovirus? I think I mentioned it a couple of times. Two-bucket disease, gastroenteritis, okay? For norovirus, the infectious dose is about 10 particles per person, which is super low, okay? For cholera, which is diarrheal disease, the infectious dose is about 1 million bacterial cells. Does that mean that norovirus is much more dangerous because it takes only 10 virus particles to establish an infection? No, it does not. Does it make sense what I just said? Or you can say, okay, the infectious dose, let's say for coronavirus is, we don't know, but probably about 200 particles. For Ebola virus, let's say also 200 particles. Does that mean that they kind of are the same? No. But if I tell you this, look, the infectious dose, I'm making numbers up, completely making them up. The infectious dose for SARS-CoV-2, the current coronavirus, is 200 particles. And for SARS-CoV-1, the one from 2003, is 50. You think, oh, it takes less particles for the first virus to establish the infection, which means the first virus got to be either more contagious or more virulent. Does that make sense? So in general, we have an association within the very close group of pathogens. We're talking about same species. We can compare within the species. We can say that higher infectious dose kind of corresponds with a lower virulence. Now this often confuses students. Just think about it. It kind of makes sense, you know. The more of something you need to cause the disease, the less dangerous it is. Am I clear? Any questions on this part?